Honey, summer has officially begun.
HIV, the human immunodeficiency virus. It's among the leading causes of death in the world, but despite its reputation and impact on the world, so little is known and understood about it. Stigma and rumors circulate around it, corrupting one's perception and understanding about the disease. I mean, where did it come from? How does it function? How does it kill? Today we know more about HIV than ever before, and it's changed the nature of how we go about treating it. In order to treat it, though, one must comprehend how it functions first. So we all remember hearing about the HIV outbreak beginning in homosexual men at the start of the 1980s, but how did the virus get into the gay population in the first place? Well, in 1999, the Hunter theory was published, which provided evidence for a hypothesis that an extremely similar virus known as SIV found in chimpanzees is the most probable ancestor of HIV. Scientists estimate that sometime around the 1920s, or even as early as the 1890s, hunters in Central Africa were exposed to the virus, either from eating chimpanzees' meat or from blood exchanges through cuts in the skin facilitated transmission of the virus. While normally the body is able to fight SIV, mutations that occurred or variations in the viral population would have allowed the formation of HIV to occur, beginning the virus's history in human hosts. Over time, the virus spreads from a hunter to the Central African population to the gay population, rumored to be the result of a rather promiscuous flight attendant, and all the way to the general population. And as the virus spreads, the titles change too, from gay-related immunodeficiency to acquired immunodeficiency syndrome, all the way to human immunodeficiency virus, or HIV. Well, if you haven't heard by now, then shame on your sex ed teachers, but HIV is a sexually transmitted infection. Vaginal or anal intercourse is the leading method of transmission from infected to uninfected individuals. As with all viral STIs, HIV functions through the exchange of bodily fluids, allowing the virus to enter the bloodstream. In addition to sexual intercourse, the exposure to the virus can also occur through blood-to-blood contact, such as through open wounds, sharing of needles, and a whole mess of other methods that are just best to avoid. Doubt this is the first you're hearing of it, but just in case the message isn't sinking in, remember, no glove, no love, and for God's sakes, just stay the hell away from needles. Once inside the cell, the real battle begins. The virus begins a series of steps in an attempt to penetrate the cell, reproduce, and spread. First, the virus must come in contact with the cell. These cells must contain CD4 receptors, otherwise cell entry cannot occur. The most popular type of cell that HIV will infiltrate is the T4 lymphocytes, also known as T helper cells. Among the numerous characteristics that make HIV such a unique type of virus, the T4 lymphocytes act as an alarm for your immune system, which then in turn facilitates its own destruction. Once bonded to CD4 receptors, secondary receptors bind, looping through the viral envelope 7 times, allowing endocytosis to occur, releasing viral components, including the RNA, into the cell, allowing infection to finally begin. Inside the cell, HIV begins a series of processes that will result in the replication of the virus. To begin, the viral RNA comes in contact with one of the other viral components, known as reverse transcriptase. If you haven't guessed by now, the reverse transcriptase will essentially work in the reverse of the traditional DNA transcription pathway. Nucleotides come to bond with the viral RNA, creating a DNA-RNA double helix. This strand is then separated by ribonuclease H, and binds again with the reverse transcriptase that finishes constructing the DNA double helix. Once the viral DNA has been formed, it bonds with the enzyme integrase, another component of the virus. This protein plays a critical role as it is responsible for the entry of the viral DNA into the nucleus, and it's integrated into the host cell's genome. This new section of DNA is known as the provirus. Now, this is virtually harmless to the cell when inactive, but after activation of the lymphocyte begins, the true dangers of HIV are revealed. Once activated, the lymphocytes begin the processes of transcription and translation. In doing so, the cell transcribes new viral RNA, which is then used to translate the proteins necessary for viral function. This RNA only codes for 9 genes too. These genes include the viral envelope, viral core, and enzymes like reverse transcriptase, integrase, and protease. Oh yeah, and protease is another key enzyme in the replication of HIV. When producing the polypeptide sequence that forms the necessary enzymes, protease comes along and essentially cuts the sequence, allowing these enzymes to form. After all this, the virus essentially has everything it needs to go off and infect other cells. Afterwards, the components pack themselves together, are encased by the viral core, and head towards the cell membrane for exocytosis. Upon their departure, they encase themselves in a portion of the host cell's membrane, taking with it viral surface proteins essential for binding to a new cell. Once the virus leaves the cell, it heads off towards a new cell to infect it, and the vicious reproductive cycle of HIV begins. Now, while that virus leaving the lymphocyte may appear identical to the original, it isn't always. All those processes of reverse transcribing, integration, transcribing, translation, and the whole rest of it leaves the virus's genetic information susceptible to mutations. This, in turn, plays a major part in the success of HIV to fight antibodies and antiretrovirals, because as these mutations occur, subtle molecular changes are made inhibiting resistance towards other strains of HIV. In terms of fighting off HIV, the human body hasn't been too successful in doing so either. In later stages of HIV, once the lymphocyte cells have detected the presence of the provirus, the body begins its attempt to fight HIV. The lymphocyte cell will trigger one of two responses. In some cases, the cell will simply undergo apoptosis as a result of the caspase-3 enzyme. Other times they will undergo pyroptosis, a process similar to apoptosis, but rather is caused by the caspase-1 enzyme and results in inflammation. This response, however, then draws in more lymphocytes, either infecting them or allowing them to undergo pyroptosis as well. This results in a positive feedback loop of lymphocyte after lymphocyte, essentially committing cellular suicide, not before the virus has already completed hundreds, if not thousands of copies. These specific processes actually went unknown for over 30 years, which now enables doctors and researchers to look at new therapies like anti-inflammatory drugs and caspase-1 inhibitors. When the virus has progressed through to this point where the immune system is destroying itself, this is where HIV transitions to become AIDS. Look at HIV as the acute infection, while a chronic infection in which the majority of the immune system has collapsed is AIDS. At this stage, the immune system has nearly destroyed itself, giving the host little, if any, protection from other pathogens. The body becomes susceptible to an array of otherwise harmless diseases, like the flu or pneumonia. Sometimes, even rare cancers can also develop, such as Kaposi's sarcoma or non-Hodgkin's lymphoma. It is the result of these illnesses that will eventually lead to death. So if there isn't a cure, then how do those infected fight the HIV in their bodies? There are actually 5 categories of HIV treatments, all of which function to inhibit crucial parts of the viral reproductive cycle. The first set of HIV drugs were reverse transcriptase inhibitors, including AZT, 3TZ, D4T, DDC, DLD, and various other acronyms that stop the production of HIV DNA sequences. Other drugs include protease inhibitors, integrase inhibitors, and entry or fusion inhibitors, all of which work by stopping one of the major steps in the cycle, but due to the high rate of mutation, the drug's effectiveness is not long-lasting. While today those infected with HIV are not automatically issued a death sentence, it is still a difficult disease to manage and combat. Individuals can expect to live for 10 to 15 years easy without the first AIDS-related illnesses appearing, so long as antiretroviral therapies are used. While we have come such a far way in the treatment of HIV, the global community still has a ways to go in fighting the disease.